As the world's population grows, it's no surprise that the conversation around living space and access to resources becomes more and more significant and complex. It's a fact that in modern times, more individuals have access to fundamental resources like running water, reliable food sources, and adequate shelter than ever before. Though the global distribution of these resources is rarely equal. But this increase in availability can come with unforeseen consequences. As the density of living in urban areas ever increases, as the size of cities and towns balloons ever larger, as more and more people inch further up Maslow's pyramid, one can't help but wonder how this affects our day-to-day behavior, our interactions and perceptions of the world around us, and the effect it has on our overall mental well-being. Some believe a series of experiments beginning in 1942 may provide a concerning glimpse into the future of overcrowded abundance, where the main limiter on population growth is not just the finite space afforded to us, but rather the limited capacity of the mind to cope with such circumstances. The individual behind the rat or mouse Utopia experiments, as they are known in popular culture, was an ethologist by the name of John B Calhoun. Calhoun began his series of experiments in the 1940s, first on rats on a farm in Rockville, Maryland, then on rats and mice at the National Institute of Mental Health, or NIM, from 1954. Retiring in 1984, but continuing his research all the way up to his death in 1995. The experiments were similar in setup, with some improvements or changes in approach and internal construction, but always with the same goal: to observe and analyze the behavior of these rat and mice populations over time, after providing them with unlimited resources, in this case being food, water, and bedding, and maintaining the populations at a high density level, but not enough to become unlivable. These experiments consistently produced the same results. The populations never grew to a size that their initial reproductive rate would imply, but rather stagnated at a far smaller number. This outcome, as well as the strange social pathologies that manifested, is what made the experiments so fascinating, and the results captured the imagination of audiences at the time, with many, including Calhoun, proposing comparisons between what happened to the experimental populations and projected effects on current and future human populations caught in similar circumstances. In this video, we'll examine the scientific papers that made Calhoun famous, his 1962 Population Density and Social Pathology, as published in Scientific American, as well as a latter paper entitled Death Squared: The Explosive Growth and Demise of a Mouse Population, published in the Journal of the Royal Society of Medicine in 1972. Calhoun begins his 1962 paper with a reference to a passage from Thomas Malthus, an influential figure in the discussion around population growth, and the author of what is now known as Malthusianism, or the Malthusian trap. As a quick aside, the Malthusian trap is the idea that populations will grow exponentially until the limitations on available resources will cause the quality of life to drop so low to trigger a mass die-off, and that this effect continues regardless of the advances in resource creation or distribution, as the population will just exponentially increase again in response to the increased availability until the new limit is reached. The thesis of Malthus, which Calhoun refers to, is regarding the ultimate limiters of a population's growth. Abstinence, that is the voluntary prevention of reproduction; misery, war, famine, disease, and the other of those dark riders; and vice. Vice here is in reference to behaviors that actively reduce population growth, rather than the prevention of activities that do so, such as abortion, infanticide, prostitution, and homosexuality. The use of contraception being seen as an immoral impossibility, with Malthus a far more enthusiastic proponent of simple abstinence. While this definition of vice, as well as the moral burden on the word, as Calhoun puts it, has certainly been updated as far as our modern attitudes are concerned, Calhoun still concerns himself with its role in population regulation, and how large an impact the social behaviors and pathologies of the individuals in a population might have. To test this, Calhoun would have to rid his experiment of the other two aspects. While he could rely on the rats to not engage in selective celibacy on ethical or moral grounds, misery provided a larger challenge. To this end, he constructed a 10 by 14 foot pen divided into 4 quadrants, each ringed by an electric fence to prevent escape or unwanted traversal. Each quadrant was a complete dwelling in and of itself, with a feeding hopper, a tray with 3 bottles for drinking, and a winding staircase leading to an elevated burrow. Each of these items was designed to maximize contact between individuals. Ramps connected all quadrants except 1 and 4, which resulted in the rats concentrating in pens 2 and 3. Each population was permitted to increase to twice the size of one that would be considered to have moderate social stress levels, and observed for 16 months. The paper describes the results of a set of 6 different populations of a particular strain of albino Norway rat. The first 3 populations began with 32 rats, and the latter 3 with 56, divided equally into male and female pairs. A year into the experiment, all populations had increased to 80 individuals, and thereafter infants that survived birth and weaning were removed in order to keep the population steady at that reasonably high density. In the first set of 3 experiments, a particular style of food hopper was used. Covered in wire grating and holding hard pellets, it made the act of feeding a lengthy affair, and increased the chance that the rats would encounter one another while doing so. It's in these populations that Calhoun first observed what he described as a behavioral sink. I'll read his description verbatim. The animals would crowd together in greatest number in one of the 4 interconnecting pens in which the colony was maintained. As many as 60 of the 80 rats in each experimental population would assemble in one pen during periods of feeding. Individual rats would rarely eat except in the company of other rats. As a result, extreme population densities developed in the pen adopted for eating, leaving the others with sparse populations. Eating and other biological activities were thereby transformed into social activities, in which the principal satisfaction was the interaction with other rats. In the case of eating, this transformation of behavior did not keep the animals from securing adequate nutrition, but the same pathological togetherness tended to disrupt the ordered sequences of activity involved in other vital modes of behavior, such as the courting of sex partners, the building of nests, and the nursing and care of the young. In the experiments in which the behavioral sink developed, infant mortality ran as high as 96% among the most disorientated groups in the population. Even in the absence of the behavioral sink in the second series of 3 experiments, infant mortality reached 80% among the corresponding members of the experimental populations. Some other notable behaviors included dominant males taking advantage of the end pens, 1 and 4, having only one entrance, proceeding to build harems and defend the walkways against interlopers, skewing the population into the second and third quadrants even more. On the occasions that males were allowed into the end territories, they would keep to themselves, not attempting to mate with any of the females, and in fact attempted to instead mount the dominant male, who would tolerate these advances. The females in the overcrowded middle pens developed an incredibly high infant mortality rate, up to 96%. They became less and less adept at building nests, and eventually gave up building nests at all. This was in part due to the constant interruptions by others during the nest-making process, forcing them to become distracted with repeated required social interactions. This also led to them being unable to move their brood with any proficiency, some infants being deposited in random locations or even out in the open, where they would soon perish and eventually be consumed by other adults. Females not in the harem pens were also constantly accosted by an overabundance of males when in heat, even the burrows not providing safety from over-enthusiastic suitors. As a result of the stress, many females died from disorders in pregnancy and parturition. Nearly half of the first and second generation females did not survive until the 16th month of the experiment, and even in the absence of the behavioral sink in the latter populations, 25% were lost. In the crowded middle pens, dominance shifts between males was constant and common, with the more aggressive males usually gaining the top spot, though the position was constantly in flux. These top males would occasionally go berserk, even attacking females and juveniles and biting them on the tail. Below these males in the hierarchy were the hyperpansexuals, who even go after other males, females not in estrus or juveniles in their efforts. These males were often the subject of accosting from the top-ranked males, but were not as active in attempting to climb the ranks. Then came the males that were completely detached from the hierarchy, ignoring rats of all sexes and even females in heat, maintaining plump figures and sleek coats. Finally, there were the probers, as Calhoun calls them. These males were absent from the dominant struggle, but were hypersexual, hyperactive, and even cannibalistic, constantly on the lookout for females in estrus, often lying in wait at the top of the ramps leading to the harem quadrants. They disregarded courting rituals, following females into the burrows and eating any dead young they found in there. It seems that even in the latter three experiments, in the absence of the behavioral sink, severe pathologies still remained, and infant mortality still peaked at 80%, an abnormally high number. It was clear to Calhoun that the Norway rat's behavioral functions either broke down or became twisted in the presence of persistent overpopulation, and he began formulating plans for more elaborate experiments, as well as a general theory around the implications of these results. The second paper, published in 1973, recounts arguably Calhoun's most famous iteration of the Utopia experiment, Universe 25. By this time, he had refined and increased the complexity of the enclosures, now featuring vertical tunnels placed in regular intervals along the high walls, connecting to food and water hoppers and regularly placed nesting boxes, creating 16 distinct cells. The hoppers also consisted of wire meshes, further encouraging the development of the behavioral sink phenomenon. Unlike the first paper, however, there were pathways between each cell, which made the creation of easily defendable harem cells untenable for the dominant males. The other distinct change was the subjects, albino mice instead of Norway rats. This time around, Calhoun retroactively broke down the development of the colony into distinct phases, A through D. Phase A was the initial 108-day period when the 4 pairs of mice were introduced to the setup, marked by considerable social turmoil as the rats got accustomed to the environment. The birth of the first litters marked the beginning of Phase B, which resulted in the exponential growth of the colony, with a doubling rate of around 55 days to around 620 individuals. Looking at the figure provided, we can see how the number of young born in each brood group during this phase is not equally distributed, despite these sections all having the identical setup. Note how moving clockwise and anti-clockwise from the southwest gives us generally diminishing returns, demonstrating the efforts on the part of the subjects to create some sort of social hierarchy, even given identical starting zones. As Calhoun notes, plotting the productivity of a brood group as a function of this ranking produces remarkable consistency. This data is also consistent with what Calhoun calls social velocity, in that the males that are more active tend to also be the more dominant in the social pecking order. This would explain the correlation between the amount of young born in each social group and the relative dominance of the males involved. At the end of Phase B, all territory was taken up by these 14 groups. Notice how there's 14 distinct groups despite the 16 separate cells, signifying a bit of a dominance overlap, each on average consisting around 10 adults, territorial male, associated males, and females and their offspring. All in all, there were 150 adults and 470 subadults and younger. This ratio is very important, as thanks to the unique setup of the experiment, there are far more subadults present than there would be in a natural environment. Phase C begins after day 315, and is marked by a much slower growth rate, with a doubling rate of 145 days instead of 55 in Phase B. This phase marks the beginning of the end for the colony, despite the continued growth rates, mainly due to the increased subadult population and lack of morbidity. As Calhoun explains, usually the number of active adults is limited by the number of social roles available, and any excess usually either perish or leave to form their own colonies. However, in this closed system, immigration and normal rates of morbidity are impossible, so we are left with a large number of individuals coming into adulthood with no open available social roles for them to fill, thus further intensifying competition among the adults. Calhoun observed that the males that failed in this endeavor withdrew, both physically and psychologically, gathering in the more neutral center of the pen and remaining inactive, apart from occasional bouts of violence upon another when they were disturbed, leaving them covered in wounds and scar tissue. The females that failed to find a role similarly withdrew, but rather to higher-level nest boxes that were unused by the active females. Naturally, with more males competing, the load on the dominant males increased as they were forced to constantly contend with fresh and desperate younger males. This lessened the ability of the dominant male to defend his territory, which caused a shrinkage of his domain. This also left many nursing females unprotected, and left them exposed to invasions of their nest sites. This caused many females to become far more aggressive than usual, taking on the defending role usually reserved for the dominant males. Unfortunately, this aggression was generalized onto their young as well, with the females attacking, wounding, and even evicting young before they were fully weaned. Similar to the first paper, the conception rate decreased, and the absorption rate of fetuses increased, as the maternal instinct became more and more disrupted, resulting in the notable decrease in the overall growth of the population that we notice in Phase C. Calhoun notes that for all practical purposes, there had been a death of societal organization by the end of Phase C. Phase D, or rather Phase Death, marks the demise of the mass population, with the increase in population suddenly halting on day 520. Then deaths very slightly beating out births till day 600, and then the incidence of successful births declining quite rapidly until the last conception on day 920. This happened due to fewer and fewer successful weans and conceptions as more and more well-adjusted females aged past menopause. The The root cause of this population death is explained by Calhoun using observations of the mice's behavior over phase C and D. By mid-phase C, almost all young had been prematurely rejected by their mothers, and had thus been unable to create adequate social bonds. These mice would then emerge into an oversized population, where their efforts to interact were not only constantly interrupted by other mice, but also forced into fragmentation, as the mice were unable to complete complex interactions between specific individuals. These factors contributed to a failure to develop more comprehensive and complicated social interactions, like courting and dominance hierarchies. From these circumstances emerged females that were unable to conceive or mate, as well as what Calhoun called the beautiful ones. Males that never engaged in sexual advances towards females, nor fought each other for dominance, thus keeping their coats pristine and well-groomed, which became the focus of their attention. As more and more of these two archetypes were born, more older subjects passed their reproductive age, the colony began to spiral into its inevitable death. As an addendum, even when samples of these archetypes were removed from the overcrowded population and placed in separate environments, being exposed to mates raised in healthy conditions, they still failed to reproduce. So, what can we take from all of this? Combining the outcomes and analysis of the two papers gives us a pretty bleak pattern, with normal social behaviors conducive to the survival of a population breaking down over time in these experimental colonies. It's not hard to imagine the impact the publication of these experiments had on public discourse around the future of high-density societal living. And the vivid, personifying language Calhoun uses really drives home the obvious comparisons that are being made here. In the latter paper's conclusion, he wonders what the effect of the erosion of social behaviors would have on human society and everyday functioning. For if my society breaks down when these basic social functions fail, then what would be the effect of such a breakdown on human societies, where interactions are far more intricate and complex and are subject to far more unexpected factors? It's here that Calhoun's critics would step in and point out that mice and rats are, in fact, not humans, which is a fair point. We certainly are several steps above in complexity. However, there is a reason that these creatures are used for medical testing. Mice and rats develop in similar ways to humans and have comparable physiologies, digestion, circulatory, reproductive, hormonal, and nervous systems, sharing over 90% of our genetic code. To write off these studies as comparing tiny furry apples to large hairless oranges would be a step too far. Other criticisms involve Calhoun's experiments not actually measuring the effects of high-density living, but rather the effects of inefficient resource distribution. After all, those males in the first paper who were able to adequately defend their harems did not succumb to the same social pathologies as the males that were unable to cement their place in the hierarchy. This is another fair point, and if we are to examine the papers again through this lens, it's not hard to see how this conclusion is reached. However, to attempt to approve or disprove this idea involves trying to argue whether or not it's possible to perpetually maintain a non-hierarchical resource distribution in such a system without excessive interference on the part of the experimenters, which might compromise the organic nature of the population's interactions and render the experiment useless. You can also imagine the political implications of scaling such conversations to humans, and that discussion has been raging for centuries unresolved. To end off then, Calhoun's experiments paint a pretty bleak picture for the future if humanity finds itself in similar circumstances as their subjects. If we will ever reach these circumstances, or if we are able to ever emigrate and expand without worrying about resource accessibility, is currently unknown. We can only hope that in the future, the mouse or rat Utopia experiments can be looked back on as a dark possibility duly avoided.